name is Natàlia Padilla. I am a postdoc at the Vall d'Hebron Institute of Research in Barcelona, Spain. In this video, I'm going to present you my poster entitled Choosing Variant Interpretation Tools for Clinical Applications. Context Matters. In the recent years, many in silico tools have been developed to help in the interpretation of variants of next-generation sequencing projects. Even though the majority of them have a high performance, none is perfect, and clinicians have to select the ones that best fits their needs. For that, performance metrics like accuracy, area under the curve, or Matthew correlation coefficient are usually used. However, these metrics don't take into account the clinical context, which may vary between countries and over time. So, to tackle this problem, we have developed two cost models, with and without rejection, that allow us to compare simultaneously an arbitrary number of pathogenicity predictors across all possible clinical scenarios. After development of this software, we have applied it to a set of pathogenicity predictors of missense variants to assess how difference in clinical context translate to differences in tool ranking. Here I show you the results of the first cost model without rejection. This is appropriate when consequences of rejection are negligible. For example, when working with proteins for which pathogenicity predictors have a very good coverage. In this case, we compute the normalized average cost of misclassification errors, applying the standard cost model, given a clinical context with this formula. Here, ρ is the proportion of pathogenic variants in the genome region sequence, and the SE and SP are the sensitivity and specificity of the predictor considered. To compare an arbitrary number of predictors and find the more cost-optimal for a clinical scenario, the normalized cost of each predictor needs to be computed, and select the one with the lowest cost. To accomplish that, we have developed a software called CSP No Rejection that divides the clinical space into regions within which only a single predictor is cost-optimal. Here you can see the application of the CSP No Rejection to the comparison of a set of pathogenicity predictors across the whole clinical space. The performance of these predictors has been assessed with more than 11,000 variants from ClinVar from the last couple of years after the publication of these methods. Here on the right, in these gray horizontal bars, you can see the predictors ranked by the area under the curve. And here on the right, in red, the results of this CSP No Rejection for two raw values. When CSP No Rejection model is applied, we can see that only 3 predictors are cost-effective in the whole clinical space: Rebel, Pompey2, and CAT. And depending on the clinical context, one or the other would be the cost-optimal one. Finally, here on the left, we can see the cost of each predictor and how these 3 become optimal in the clinical space. Additionally, we can also extend this model to take into account the rejection, that is, when predictors do not produce an output. In this case, the formula is this one, with alpha, that is the coverage. The set of RC0 and RC1 pairs comprises all possible clinical scenarios, when misclassification and rejection costs are considered. This set defines a triangular region, that we call clinical space, and it can be used to systematically rank predictors in multiple clinical scenarios in terms of cost. For that purpose, we have developed the CSP rejection algorithm that divides the clinical space into regions within which only a single predictor is cost-optimal. Here in Figure 2, you can find the results for that set of pathogenicity predictors and variants, now taking into account also their coverage. The comparison of all possible pairs of predictors results in this set of lines, which divides the region into these polygons, where a single predictor is cost optimal. After the merging of polygons with the same optimal predictor, we can find these large colored regions, where a predictor is cost optimal. Finally, we can compare the fraction of these cost scenarios, where a predictor is optimal, and the ranking of these predictors by the area under the curve. To finish, I just want to mention that we also, in this poster, I show you the study of the raw parameter to explore the dependence of the previous ranking of predictors on the proportion of pathogenic variants in the genome region sequence, and a case study of the TP53 protein. Thank you for listening.